Welcome, fellow denizens of the dark. This is Lance, your host of Strange Bites, where we sink our teeth into wonderfully weird and wild science discoveries. I really appreciate everyone tuning in. If you enjoy the show, please hit that subscribe button on your favorite podcast app, leave a quick review, and follow us on social media. Your support truly makes a difference. Now let's dive into today's bite. Imagine you lose a finger in an accident. The wound closes, a scar forms, the story ends there. A permanent reminder etched in your flesh. Now imagine something else. The same wound, but instead of a scar, new bone grows. A joint reassembles. Tendons and ligaments weave themselves back into place like they were following an ancient blueprint. You don't just heal, you regrow. For most of human history, we believed this was impossible for us. Scarring was our destiny. Regeneration belonged to myths, to salamanders, to creatures from another world. But what if the power was never gone? What if it was only switched off? In episode 36, we dive into a real scientific discovery that just flipped that switch in the lab, and what it might mean for every one of us. Stay with me. This one gets under the skin in the best possible way. Today's story comes from the labs of Texas A&M University, where researchers have done something that sounds like science fiction, but is grounded in cold, hard data, published in the journal Nature Communications in April 2026. On one side of the evolutionary stage stand the salamanders, the axolotls especially. These smiling, feathery, gilled wonders can lose a leg, a tail, even parts of their heart or brain, and grow it all back perfectly. No scars, no loss of function. They carry a kind of biological magic we've long envied. On our side, us mere mammals, we scar. When tissue is damaged, special cells called fibroblasts rush in like emergency contractors. They lay down dense, fibrous scar tissue to seal the wound fast and prevent infection. It works. It saves lives. But it also locks the door on true rebuilding. The fancy structures, organized bone, working joints, gliding tendons never come back the way they were. For generations, scientists asked, did we evolve away their ability to regenerate? Or is it still buried somewhere inside us? Just silence. Enter Dr. Ken Munioka and his colleague Dr. Larry Suva at Texas A&M's College of Veterinary Medicine and Biomedical Sciences, along with a team spanning Tulane, Stanford, Arizona State, and even Vienna. They didn't add magical stem cells from outside the body. They didn't rewrite DNA. They simply asked the body's own cells a better question, in the right order, at the right time. Here's how the story unfolded in the lab, told as plainly as I can make it. They worked with mice and focused on tiny but telling injury, amputating the tip of a digit, the last bone segment. In adult mice, this normally heals with a blunt stub and scar tissue. No regrowth. Classic mammalian response. But the team tried something new, a precise two-step sequence using two well-known molecular signals that cells already understand. Think of them as text messages or keys that unlock specific behaviors. First signal, FGF2, fibroblast growth factor 2. They applied it a few days after the wound had already started its normal healing process. Not too early, not too late. FGF2 whispered to the fibroblast already at the scene, hey, instead of just patching with a scar, why don't you gather and organize? Remember how things were built in the first place. The result? The cells began forming something that looks like a blastema, a bud or mound of versatile rebuilding cells. This is the same kind of structure salamanders use as their regeneration headquarters. In mammals, it almost never happens after an injury. Here it did. The wound cells didn't need brand new stem cells imported in. They were already there, capable, just waiting for the right nudge to shift from scar mode to rebuild mode. Timing mattered enormously. Apply FGF2 too soon or without the follow-up, and the structure might form but then fade away. The cells needed the first push to change their behavior. Then came the second signal, days later. BMP2, bone morphogenic protein 2. This one said, now build, differentiate, turn that gathered potential into actual structures. And the body listened. New bone began forming where the amputated tip had been. A growth plate appeared, a feature straight out of embryonic development, suggesting the process was tapping into ancient instructions. Joint structure started reassembling. Tendons and ligaments from the remaining stump contributed to a more complex repair. Even a small seismoid-like bone, a little extra bone that helps with movement, showed up in some cases. It wasn't perfect. The new digit wasn't an exact flawless copy of the original. There could be some extra bone in odd places or slight imperfections in the shape. But it was real regeneration. Organized skeletal elements, connective tissue, and joint-like structures where before there had only been a scar. Multiple major components that had been removed grew back. The researchers showed that two different responses worked together. One blastema dependent for the main bone tip, and a blastema independent path for the joint complex. Crucially, cell lineage studies revealed something profound. The wound cells that entered this regenerative process were being positionally respecified. In simple terms, they got new instructions about what they should become and where they belonged. It's like the cells had a map and were told, you're not just genetic repair material anymore. You're the tip of a toe again. Dr. Munioka put it beautifully. Regeneration in mammals isn't absent. It's just obscured. The capacity was never truly lost. The right signals delivered in the right sequence can rescue it. And here's the part that makes this feel almost alchemical. They didn't need exotic new technology or external stem cell transplants. They used two proteins the body already knows. Growth factors that play roles in normal development and healing. BMP2 is even already FDA approved for certain bone-related medical uses. FGF2 is in clinical trials for other applications. That lowers the barrier to someday translating this into real therapies. The discovery, published as Digital Regeneration in Mice, is stimulated by sequential treatments with FGF2 and BMP2, proves that at a normally non-regenerating wound site, the right molecular conversation can flip the script from scarring to rebuilding. Let that sink in for a moment. The same kind of cells that normally rush to create a messy but effective scar can, under the right instructions, organize into something far more sophisticated. The construction crew was always on site. We just needed to give them better blueprints and tell them the job isn't finished when the hole is plugged. This isn't yet a cure for lost limbs in humans. It's early stage work in mice, focused on small digit amputation. Scaling it up, making it perfect, ensuring safety in larger bodies and different injury types. That's the long road ahead. But it's a genuine crack in what we thought was a sealed door. A proof of concept that mammalian regeneration can be coaxed back into existence. Now, as we always do here on Strange Bites, let's step back from the lab bench and stare into the deeper implications. Because this isn't just about fixing toes, it's about what it says regarding the nature of life, injury, and what it means to be human. If our bodies already carry the latent machinery for regeneration, if the difference between us and a salamander is partly just which signals are active after injury, then many of the hard limits we accept about human frailty might be softer than we believed. Scarring evolved as a survival advantage, quick and dirty, seal the wound fast, fight infection, live to see another day. But in doing so, we traded away the slower, more ambitious path of true rebuilding, This study suggests we might not have to choose, or at least we might be able to nudge the balance. Think about the future this opens. In medicine, imagine a world where severe injuries or surgeries don't leave behind stiff, painful scar tissue that limits movement or causes chronic problems. Heart tissue after a heart attack could potentially repair more functionally instead of forming scars that weaken the pump. Spinal cord injuries or nerve damage might see better reconnection. Lost digits or even more complex structures could one day be coaxed back. The quality of life after trauma could transform. Combined with other advances in biomaterials, gene therapy, or precise delivery systems, might move us from managing damage to restoring wholeness. But the philosophical questions are even richer and stranger. What does it mean for our relationship with our own bodies if perfect repair becomes possible? Scars have always been part of our stories, badges of survival, maps of experience, visible proof that we lived through something. If those marks fade or never form, do we lose a certain kind of narrative depth? Or do we gain freedom from unnecessary suffering? More existentially, if we can flip biological switches that evolution left dormant, are we playing God? Or are we simply remembering instructions that were always written into our cells? The researchers' work shows that regeneration competent cells exist at the wound site and can be redirected. That blurs the line between what is natural and what is possible with the right key. It echoes older ideas in philosophy and mysticism about latent human potential, the idea that we are more than our current expression. On the cautionary side, power like this demands wisdom. Who gets access? Will regenerative therapies be available to everyone, or will they widen existing inequalities in healthcare? What unintended consequences might arise from overriding the body's default scar and survive programming? On a large scale, could overzealous use change how we experience aging, risk-taking, or even identity? And deeper still, if injury and imperfect healing have shaped human culture and even art, think of all the stories born from wounds and scars. What happens when loss becomes more optional? Yet the dominant feeling this discovery leaves me with is wonder, not dread. It's a reminder that life is far more plastic and surprising than our everyday assumptions allow. The same biology that gives us scars also contains the seeds of something more ambitious. We are not fixed, finished machines. We are ongoing processes, and sometimes, with the right nudge, those processes can remember older, more powerful ways of being. This research doesn't promise immortality or effortless regrowth tomorrow, but it does something more profound. It shows that the door was never fully locked. The switch was only hidden. So the next time you look at a scar on your skin and feel that quiet resignation, this is just how healing works. Remember the mice in that Texas lab. Remember the two precise signals that said, "Not today. Today we build instead." Our bodies still have secrets to reveal. Science keeps showing us the limits we accept are often invitations to look closer, experiment more carefully, and ask better questions. What other hidden switches are waiting inside us? What other impossible things might simply need the right sequence, the right timing, and the courage to try? That's it for today's bite. Let it linger. Let it make you question what else we've accepted as final. If you enjoyed the story, hit that subscribe button on your favorite podcast app. Give me a follow on social media. And until next time, stay strange. Question everything. And keep wondering about what your cells already know.